Welcome to the lecture series in food microbiology. In the last couple of lectures, we have been seeing what are the illnesses which are caused by the consumption of certain food products, the causative organisms which cause these illnesses, how do we detect their presence or the presence of the toxins in the food, as well as what are the steps which we take to eliminate these organisms or the products so that the food poisoning or foodborne illnesses can be avoided. We will be discussing more about these in today's lecture also. In today's lecture, I'll be talking about a major foodborne organism which causes disease. One of the causative organisms is actually very lethal, Clostridium botulinum. The genus Clostridium belongs to gram-positive spore-forming organisms. They are rod-shaped organisms. These cause the bacterial food poisoning. And among the gram-positive spore formers, you have three main organisms which we are concerned with: Clostridium perfringens or Welchi, Clostridium botulinum, and Bacillus cereus. We already dealt with the intoxication caused by Bacillus cereus in a previous lecture. Clostridium perfringens. This is known to be the causative organism of a serious wound infection, which was called as gas gangrene in 1892, when it was first described by the American bacteriologist Welch. So initially, it was a surface organism. It was the foodborne relationship of this organism, and that it can cause foodborne illnesses was not earlier known. The first known episode of this causing a human disease was called the causative organism as a gas gangrene. In the mid-1940s, the outbreaks associated with school meals in England and pre-cooked chicken dishes in the USA firmly established that Clostridium perfringens is a cause of food poisoning. Based on their ability to produce certain exotoxins, 5 types are recognized: types A, B, C, D, and E. And the food poisoning strain belong to type A. The pathogenesis and the disease symptoms of several pathogens associated with foodborne and waterborne gastroenteritis are somewhat different from the classical food poisoning or foodborne infection. Gastroenteritis, which is caused by Clostridium perfringens, Bacillus cereus, the Vibrio cholerae, and their enteropathogenic E. coli, we call it as a toxico-infection. What are the characteristics of a foodborne toxico-injection? For one thing, if it is a spore former, you have to consume or ingest a large number of live vegetative cells. The vegetative cells of these spore formers, they do not multiply in the digestive tract, but they sporulate in the digestive tract and release the toxins. In the case of gram-negative bacteria, like E. coli, enteropathogenic E. coli, live cells can be ingested in moderate numbers. These gram-negative live cells which have been ingested will then rapidly multiply in the digestive tract, causing the disease. And many cells also die, releasing toxins. And toxins of both groups, whether it is gram-positive or gram-negative, they cause the gastroenteritis symptoms. In the case of Clostridium perfringens, they are gram-positive rods, motile, and spore formers. The cells vary in size and can form small chains. This is anaerobic, but to some extent it can tolerate air, rocks, or oxygen. The vegetative cells of Clostridium perfringens are sensitive to low heat treatment or otherwise pasteurization, but the spores are extremely heat resistant, and some can even survive boiling for several hours. Spores actually, as you know, are formed when the organisms are under adverse environmental conditions. So they are a survival mode for these organisms. So when in a survival mode, it is going to be very difficult to kill them. And these cells are resistant to D-cycloserine. In the presence of suitable substrates, hydrogen sulfide is formed during growth. The cells need several amino acids for growth. And since these organisms require amino acids for growth, many proteinaceous food substances can be an ideal nutrient medium for the growth of these organisms. In nature, the spores as well as the vegetative cells are found in soil, dust, intestinal contents of animals, birds, humans and sewage. Many foods, especially raw foods, get contaminated from these sources. This blue structure is actually the spore inside the organism. Among the 5 types of Clostridium perfringens, type A strains are predominantly involved in foodborne toxico-infection. The toxin, or otherwise the enterotoxin, associated with this foodborne disease is a heat-labile protein. It is an intracellular protein produced by the cells during sporulation in the intestine, and then it is released. Unlike the toxins of food poisoning organisms, the enterotoxin is produced inside the digestive tract. The environmental parameters for the production of enterotoxin are directly related to the sporulation environment. The Clostridium perfringens type A, which is responsible for food poisoning and gas granulin, produces an alpha toxin. This alpha toxin has a lecithinase or a phospholipid seat activity. So, as the term indicates lecithinase, it is capable of hydrolyzing lecithin. It is also capable of hydrolyzing phospholipids. So, this capability actually plays a huge role in the pathogenesis of gas gangrene. By attacking the cell membranes, it produces local tissue disruption in the wound, and then its absorption into the circulation causes the serious toxemia. Clostridium perfringens type C, which produces both alpha and the beta toxins, causes enteritis necrotoxicans, a more severe but a very rare and a serious enteric disease in which the beta toxin damages the intestinal mucosa, causing necrosis. So here, the alpha and the beta toxin, especially the beta toxin, causes the intestinal mucosa to degrade or undergo necrotic damage. So the cells apoptose. Illness is preventable by the active immunization against the beta toxin. Symptoms of abdominal pain, bloody diarrhea develop several days after consumption of a high protein meal, usually pork consumed on festive occasions. Low levels of intestinal proteases are predisposing factor in victims. Type A heat-resistant strains produce a theta toxin. This is otherwise called as perfringolysin O or PLO. This is similar to the thiol-activated hemolysin, similar to listeriolysin O, which is produced by Listeria monocytogenes. Clostridium perfringens food poisoning is generally self-limiting. It is a non-febrile illness characterized by nausea, abdominal pain, diarrhea, and less commonly vomiting. The onset of the disease or the symptom is usually 8 to 24 hours after consumption of food containing a large number of the vegetative organism. Once these vegetative organisms get into the GI tract, they survive the stomach's acidity, pass to the small intestine where they grow. And inside the small intestine, they sporulate and release the enterotoxin. The enterotoxin is synthesized by the sporulative cells, although low level of production has been seen in the vegetative cultures also. Normally, the enterotoxin is synthesized only by the sporulating cells, but vegetative cells are also capable of the production of low level of these enterotoxins. And normally this toxin is closely associated with the spore coat. And the toxin is released into the intestinal lumen on the lysis of the sporangium. This enterotoxin is a 35 kilodalton protein with an isoelectric point of 4.3. This toxin can be inactivated by heating in saline at 60 degrees centigrade for 10 minutes. It is sensitive to certain proteolytic enzymes also. So the activity of this enterotoxin is like the cholera toxin. It gets bound to the intestinal epidermal cells. It reverses the flow of sodium, chlorine and the water across the gut epithelium from absorption to secretion. So normally the epidermal cells or the lining of the intestinal mucosa is capable of absorption. But here, because of the binding of the toxin to the epidermal mucosa, it releases or reverses the ionic flow, causing water to outflow to the intestine, causing the onset of diarrhea. It first binds to specific protein receptors on the epithelial cell and is then inserted into the cell membrane, producing morphological changes within a few minutes. This changes the cell permeability, inhibits the synthesis of cell macromolecules, and then produces pores in the membrane of the cell, which eventually dies as a result of the membrane damage. So ultimately, the cell will die because of the damage caused to the cell membrane. How do we diagnose this? Diagnosis of Clostridium perfringens food poisoning is normally based on a number of factors. One is the case history and symptoms, meaning what type of food did you have, what is the sort of symptoms produced by the patient. The large numbers, which is greater than 10 to the power of 6 per gram of Clostridium perfringens pores in the patient's faces. Large numbers of the vegetative cells of the same serotype in incremented food, and the presence of the enterotoxin in the feces. So all these four will give proof that the food poisoning is due to Clostridium perfringens. Raw meat from animals and birds is the most common contaminated with the spores, and cells from the vegetative tract content, whereas vegetables and spices commonly get them from soil and dust. As the bacterium has amino acid requirements for growth, meat and meat products provide good environments for the cell growth. The common foods associated with outbreaks include meat stews, especially beef and poultry, roasts, meat pies, casseroles, gravies, sauces, bean dishes, and some Mexican foods like tacos and enchiladas. Isolation and identification. Once the organism has been isolated, you require normally enrichment. But in this case, one of the causative reasons would be the isolation of a huge amount of organism or the presence of a huge amount of these organisms in the feces. So enrichment culture is rarely necessary since Clostridium perfringens will invariably be present in high numbers both in the implicated foods as well as in the clinical samples. On the examination of the food, the total count, that is vegetative cells plus the spores, this is determined, but with the fecal specimens, a spore count is obtained after heating a suspension at 80 degrees centigrade for 10 minutes. This is also performed. The commonly employed technique is selective plating. In selective plating, we use antibiotics and sulfite reduction to produce the black colonies as the differential reaction. So if these black colonies are present using antibiotics and sulfite reduction, this confirms that the organism which is grown is Clostridium perfringens. And the most popular combinations in plating is tryptose sulfite cycloserine medium and the olandomycin, polymyxin, sulfadioxin perfringens incubated anaerobically for 24 hours at 37 degrees. And in the pour plates, white colonies appear on the agar surface. Suspect colonies can be confirmed by the absence of motility, their ability to reduce nitrate to nitrite, the lactose fermentation, and gelatin liquefaction. A traditional confirmatory test, the Nagler reaction, which looks for the production of the alpha toxin and its neutralization by the antitoxin on the lactose egg yolk agar, is less favored now because of its low specificity and the occurrence of lecithinase negative strains. Normally, this confirmatory reaction where we look for the presence of the alpha toxin, and if you add antitoxin to the medium, you will see a toxin-antitoxin complex surrounding the bacterial colony on lactose egg yolk agar. This gives positive confirmation for the presence of the organism. But the problem is the specificity for this test is low and you have lecithinase negative organisms also which are present. So this test is not used these days. Serotyping based on capsular antigens is employed for epidemiological purposes. A commercially available kit employs reverse passive lactase agglutination. The name derives from the fact that in a standard agglutination assay, soluble antibody reacts with the particulate antigen such as the bacterial cells. And in reversed passive latex agglutination assay, the soluble antigen will react with the antibody attached to the lactase particles. These latex particles play no part in the reaction and they are passive, but they do provide a visual signal when they cross-link as a result of the antigen-antibody reaction. So visually, you would be able to see the positive presence of the organism, indicating the positive reaction. Clostridium perfringens uses its large arsenal of protein toxins to produce histotoxic, neurologic, and intestinal infections in humans and animals. So, a large number of toxins which have varying effects like histotoxic, the neurologic, and intestinal infections are caused by these toxins. The major toxins involved in diseases are the alpha, otherwise called as the CPA toxin, the beta, which is called as the CPB toxin, the epsilon, which is called as the EDC toxin, then the ITA or the ITX, the enterotoxin, which is CPE, and the necrotic B-like or the net B toxins. So this arsenal, these are the arsenals of toxins which are synthesized by Clostridium perfringens. CPA is the main virulence factor involved in gas gangrene in humans. Its role in animal diseases is limited and it's controversial. CPB is responsible for necrotic enteritis and enterotoxemia. This is mostly in the neonatal individuals of many animal species, including humans. ETX is the main toxin involved in enterotoxemia of sheep and goats. ITX has been implicated in the cases of enteritis in rabbits and other animal species, but whether it has a role in causing disease has not been proved yet. CPE is responsible for human food poisoning and as well as non-foodborne Clostridium perfringens mediated diarrhea. So this is CPE which would cause the human food poisoning. Net B is the cause of necrotic enteritis in chicken. And in most cases, the host toxin interaction starts on the plasma membrane of the target cells via specific receptors, resulting in the activation of intracellular pathways with a variety of effects, commonly including cell death. So what happens is that the toxin will go and get bound to the receptor on the plasma membrane. And this receptor is specific. So it is a specific receptor, specific reaction. The toxin is compatible with only specific types of receptors. So once the receptor is bound with the ligand, in this case the toxin, this will accelerate or activate a series of reactions, intracellular pathways. This series of reactions in turn will lead to effects on the cell. The most lethal is the causing cell death. In general, the molecular mechanisms of cell death associated with Clostridium perfringens toxins involve apoptosis, necrosis, and necroptosis. Perfringens is the most commonly isolated Clostridium species, and 5 types of toxins A and D may be distinguished based on the production of the major lethal toxins. Like we said, indicated before, A to E, you have the 5 types of toxins, all secreted by Clostridium perfringens. There is another more famous species in this, Clostridium tetani. This produces 2 exotoxins, the tetanolysin and the tetanospasmin. The tetanolysin causes lysis of the RBCs and serves no known benefit to Clostridium tetani infections, while tetanospasmin is a neurotoxin. This tetanospasmin is what causes the clinical manifestation of tetanus. Another species which is foodborne and lethal is Clostridium botulinum. This also produces neurotoxins. The botulinus toxin is the most potent natural poison ever known. This causes botulism, a disease characterized by symmetrical descending paralysis. Here we have 7 toxin types, A to G. Human is susceptible to A, B, E, F and G toxins. Types A, B, C and D cause intoxication in animals. So Clostridium botulinum, which we'll be discussing later, this produces neurotoxins with 7 categories, among which A, B, E, F and G can cause harm to human beings. A, B, C and D can cause intoxication in animals. Clostridium difficile. This is also a most common toxigenic Clostridium species. Produce 2 potent exotoxins, toxin A, which is an enterotoxin, and toxin B, which is cytotoxic activity. So 2 toxins are present here in the arsenal, an enterotoxin as well as a toxin with cytotoxic effect. You have another Clostridium species, Nui, which comes with 3 types labeled A, B, and a non-pathogenic type C. This is also distinguished by the range of toxins they produce. Clostridium sordeli. This produces 3 toxins in common with the non-pathogenic Clostridium bifurmentans. This also has a major lethal toxin called as the beta toxin. And the presence of this beta toxin actually distinguishes it from Clostridium bifurmentans. To distinguish between Clostridium sordeli and Clostridium bifurmentans, we look for the presence of beta toxin. If there is beta toxin, it is sordeli. If there is no beta toxin, then it is Clostridium bifurmentas. And this beta toxin contains 2 subcomponents or 2 toxins, a lethal toxin which is called as the LT and a hemorrhagic toxin which is called as the HT. As the name indicates, this can cause hemorrhage. The detection of these organisms or the presence of Clostridium, commercially we have a large number of detection kits which are available. For example, for the detection of Clostridium botulinum, this is called as a Lyo kit, marketed by Biotecon. Then you have the Clostridium difficile test kit, marketed by the Norgan Biotech. You have the Clostridium difficile, again a rapid diagnosis kit, which is again marketed. You have the Clostridium perfringens alpha toxin detection kit marketed by BioIngentech. So all these kits, commercially available kits, can be used to detect the presence of either the organism or the toxins which are present in food. So food as well as suspected food or samples from the clinical samples from the patients can be used as a substrate for the testing for the presence of these organisms using these commercially available kits. The genus Clostridium, as we saw, contains a huge number of species which are potential pathogens. The implication of these as a pathogen is varying, and in many cases even very low numbers are capable of producing symptoms and causing illnesses. So the early detection as well as isolation of these organisms in food becomes very important. The commercially available kits can do this purpose. We have a variety of kits which are available, each which do either PCR based or ELISA based or protein isolation with the tests to identify the presence of these toxins, not only from human samples but also from food samples. The other major organism which we will be dealing with in this case is Clostridium botulinum, which produces neurotoxin. This will be separately dealt with in a subsequent lecture. In this lecture, we saw about the identification techniques for Clostridium perfringens and how the action of its toxin manifests. If you are given a food sample, say a vegetable sample versus a meat sample, what would be the protocol which you would use to isolate or identify the presence of Clostridium in this? I am looking for both the vegetative cells as well as the presence of the toxins. What methodology would you use to isolate? What sort of a medium would you use to grow these organisms? And then, what is the step which you will take to isolate the protein from the sporulating organism or from the contaminated food? Finally, to avoid the presence of either this organism or these toxins, what would be the step which you take? What sort of treatment would you give? Here you have to remember that the spores are extremely resistant. And as we have seen earlier, spores can be destroyed only under extreme high temperature and pressure, which we normally use in autoclave. So to destroy a food which contains spores, then you would have to use that much amount of pressure and temperature, which in turn can be unsavory because the texture or the nutrient component or the taste of the food or the flavor of the food might go off. So in this scenario, in food processing, how will you compromise? This is something which you have to think about. To summarize, we saw that the genus Clostridium is capable of causing a series of foodborne hazards The lethality depends upon the type of toxin which is synthesized by the sporulating organism. The detection of this toxin is extremely important, or toxin as well as the organism. We will be dealing more with about the damage caused by the other strains in the subsequent lecture.